And we'll do one tomorrow morning, and tomorrow morning we will also do the workshop. So I trust that you have all received the files for the workshop. Sent in last week, I think. So what I would please ask you to do is to put in your answers on the PowerPoint file. So in the bit where you have to highlight the primer sequence, etc., if you could just highlight that on the sequence which is in the PowerPoint file. The same with the last bit where you have to put in the PCR band and the restriction fragment polymorphism, restriction fragment bands. If you could also just put them in on the gels which is shown in the PowerPoint file. And what I then will ask you to do, because I won't give you the answers to the workshop, you have to show me your answers to the workshop. And if you put them in on the PowerPoint, that means that you can then share the file with me, and you can show me and the rest of the class what you got. Okay? So be prepared. Please go through the workshop and put your answers into the PowerPoint. Okay? Because I'm expecting you to give me the answers tomorrow. I am not giving them to you, okay? Okay, so let's start with the first part of the Human Genome Project. Okay, so let me go on to full screen here. In a minute, just move that. There we go. Okay. That should now be on full screen, so let me just get all of these other things out of the way so I can actually see my screen. Okay, so as I've already said, today and tomorrow, the only reason, you could have done them all today, but I do know that it is quite difficult online to keep your concentration for too long, particularly for lectures where it's just me talking and you not saying or doing very much, but really just listening. So I do know that that's quite a challenge, and it can be quite difficult to keep your concentration for too long. So we'll do the first part, we'll do the first lecture today, and we'll do the second lecture tomorrow. Okay. So obviously today and tomorrow we're going to talk about the human genome and the sequencing of the human genome. So today we'll talk primarily about how it was done, who did it, etc., etc. And tomorrow we will look more at what have we actually learned from the Human Genome Project. So what have we learned from now having the sequence of the human genome? So here are your learning outcomes. So this is the learning outcomes both for this lecture today, but also for the lecture tomorrow, okay? So your learning outcomes is really to appreciate the motives behind the Human Genome Project. And you should understand, so this is important, that there is two major methodologies of which were applied. So that will make more sense in a minute. You should also know the major findings of the Genome Project and realise the area in which the Genome Project have been practically applied. So here are some of these practical applications. So there's been, after the human genome was sequenced, and people actually realised the genetic variation in the genome. There has been quite a few large-scale projects which have used that genetic variation or looked further at characterizing that genetic variation between people, and used it, which you'll hear more about in next year's Bm210 really, where you will hear more about how single nucleotide polymorphisms is used in genome-wide association studies, looking at a variety of different diseases. We won't talk much about that this year. Okay, from the Genome Project, we've also realized the nature of the genetic differences in the population of the world. So population genetics, so how Europeans differ from Asian, etc., etc. Those genetic differences that can be found by this between different populations in the world. The role of natural selection and evolution in human genetic variation. And we're also going to look at pharmacokinetics. So really understanding how drugs or how genetic can affect how drugs are metabolized. And also we'll look a little bit at the methods that are available to speed up human genetic research. Okay, so let's start off with what is a genome. So one genome is obviously the complete set of chromosomes. So you already know all this. You know that we've got 23 set chromosomes. One is obviously the sex chromosomes, as you can see here, X and Y. So one genome is really the whole complete set of these chromosomes. The human chromosomes, as you can obviously see here, do range in size. So from about 55 million base pairs to 250 million base pairs. And the biggest chromosome, as you can see here, is chromosome 1. And then you can see that the size of the chromosome is decreasing here in the descending order. So the smallest chromosome, as you can see here, the Y chromosome. And also what we have already talked about. So this is kind of leading up to a lot of what you're going to hear today. But what we have talked about is that each and every genetic, so remember there's genetic maps and there's physical map. And remember that each of these genetic and physical markers, so we use these markers to make these maps, each and every genetic and physical marker has a specific locus in the genome, okay? So it's really just a location. It has a specific location in the chromosome. Somebody's joining us. There you go. Okay, so the goal of the Human Genome Project was really, or the justification that they gave to the funders for the project was obviously to determine the sequence of the about 3 billion chemical base pairs in the human DNA. And what they wanted to do after it was sequenced was really to identify all of the genes in the human DNA and their position. So where and on what chromosomes were these genes. They also wanted to attempt to predict the function of all genes. And obviously that's very useful information because understanding the function of all genes, that information can obviously be utilized for understanding diseases. And understanding diseases helps us to develop better medicines. Obviously, it's also very useful in DNA forensics. It helps us also to understand the differences or the variability between humans and how does human compare to other species. So that's us back to evolution, really. So the evolution of humans' DNA and the function of our genes are very much important. There's a couple of websites down here. You can go and have a look at them yourself in your own time. But there are some good websites to go in and look at the Human Genome Project. So this here shows you a timeline, as you can see. So the Human Genome Project was initiated by the Institute of Health in the United States. So although it was a collaboration between many people in the world, it was initiated and led by the National Institute of Health in the United States. And it was initiated, not people coming, it was initiated originally by this guy here, which is called James Watson in 1989. The name might very well be familiar to you because he also received a Nobel Prize for being involved in determining the structure of DNA. So you have probably heard his name before. So it was initiated by James Watson, who was the director of the National Institute of Health. However, it was taken over by Francis Collins. So you can see here that in the early 90s, Francis Collins became the director of the National Institute of Health because James Watson really retired. Now, if you look at the timeline here, you can see that before, or a lot of the activity really in the Human Genome Project was this, so you can see that in the orange boxes here. So it was the genetic and physical mapping of the genes, okay? So genetic and physical mapping, it really is what took up the majority of the time, as you can see here, because once they actually starts to sequence, so you can see here that the pilot sequencing is starting in 96, and really within 3 to 4 years or so, we do have the first full-scale sequencings really begin. So in here between the piloting and the full-scale sequencing, very much have to do with improving technologies, etc., to get high throughput, really sequencing technology. We'll talk a bit about sequences technology and the improvement in that a bit later on in this presentation here today. But you can see here when they actually start this full-scale sequencing of the genome, it doesn't really take very long before there is a draft of the genome. So really the majority of time is spent on these genetic and physical maps. So we've already talked about these. But it took about 13 years to get a draft sequence of the human genome. What you can also see here is that in between, so to get practice, the sequence, so you can see here we've got the yeast, E. coli, C. elegans, and the DNA of the cephalothri is sequenced here in the beginning of the 2000s, as you can see. So really the initial goal of the Human Genome Project was very much to build these genetic and physical maps of the human and other genomes. And you can see that they initially sequenced these smaller yeast and worm genomes, and that was really very much like a test run for sequencing this much larger and much more complex human genome. Oops, we're not moving forward. Now, there was some unexpected competition to the Human Genome Project led by the National Institute of Health. And that unexpected competition came from a private company which was led by this guy called Craig Venter. The company was called Celera. Now, what Celera wanted to do was also to sequence the human genome, and they aimed to sequence and assemble the entire human genome by 2001, which is what you can see in this timeline here. So you can see that this project is initiated, first of all, a lot later. Okay, so we've got public funding agreed here in the early 90s. Okay, so that is when funding is awarded and the National Institute of Health initiates the whole Human Genome Project. And you can then see here that much later, much later, so really not until the late 90s here, Craig Venter, who owns this private company, Celera, starts his Human Genome Project. Okay, so that's in 98. So within 3 years, they have a target completion date. So much faster process, much quicker than the process we're looking at for the National Institute of Health. Now, the biggest difference between these two projects was that the National Institute of Health, which you'll see on the next slide, so the Human Genome Project very much was a project which was led by the National Institute of Health. However, it was a collaboration between many, many partners all around the world. So many labs around the world was actually involved in sequencing the human genome. And also, in the National Institute, in this publicly funded genome project, the aim was very much, or the agreement really was, that the sequences had to be made available to the world, basically, to anyone, not only to these labs, but to any member of the public within 24 hours of the sequences. So it should be freely available. Everyone could go in and see these sequences as they emerge. Now, that was not really the philosophy of the privately funded project, because what Craig Venter wanted, he wanted to earn some money on this sequencing. So he was really planning to file patents on genes and before releasing these sequences. So very different philosophy to the one for the National Institute of Health. Okay, so this is just showing here that although the project was initiated by the United States, it very much was a very formal international consortium which participated in the mapping and the sequencing process. So there was really formal agreements between the many labs across the world which was involved in this mapping and sequencing process. So you could see here that individual labs very much concentrated on single chromosomes. So there was labs in the United States, the United Kingdom, Germany, China, and Japan are just some of the ones which I mentioned here. Now, before they could really start the sequencing process, what was required was the ability to build these robotic production lines that could carry out these very large number of PCR tests and sequencing reactions that was required to sequence the whole of the genome. So there was really quite a lot of technological advancement in those 10 years as well. And then, as I've already said, the idea then was, or the agreement was, that the information, so every bit of information, sequencing data that came out from this project was published and it was shared via the World Wide Web. Okay? So really important in this agreement that all data are available to not only the labs involved in this process, but really any individual who was interested in obtaining the information. Okay, so there's different phases to the project. So phase one, already said that really, was to produce these high resolution chromosomal maps. Okay, so it's positioning these genetic markers first of all, so that we can position the genes within the genome, and then after that create these libraries of back clones that could then be sequencing. So this is us onto the physical map. Okay, so we start off with the genetic markers, and the genetic markers will allow us to position where in the genome you are. you then got to translate that genetic map into a physical map. And the way you do that is by cloning. So remember, to clone, you chop up all this chromosomal DNA and you then put it into these bacterial plasmid clones, okay? And that's how you can then increase your yield. So first of all, you will obviously, these bacteria will proliferate and you'll select. Remember, we've got the antibiotic selection gene in these clones, and based on that, we can then select all of the bacterial clones which have got the plasmid that contains what it is that we want it to contain, etc. So that's, you have to think back to the previous lecture that we did last week to understand this really. So go back, if you don't know what physical map is, go back and have a look at that lecture. Now, once you have this physical map, you then need to start to sequence each of these back DNA clones. So remember, these genetic and physical maps involves making these contigs, so you will have a lot of overlapping clones. And, you know, you order these clones into a contig, and then you start to sequence them. It's ordered into these contigs based on the position of these genetic markers. Okay, so you know the locus of these genetic markers, and that is how you can order it into this contig. Now, once it's then sequenced, you then need to obviously assemble all these sequences. So you've sequenced all of these backbone, and then you need to assemble that into a sequence. And then once you have all these sequences, then you can produce a final draft and annotate to identify the genes within the genome. Okay, so this shows you an overview of how. So I want to really stress the fact that the way and the approach that the National Institute of Health, so the public project, compared to the privately funded project, so the project that Craig ventured on, the approach to how, not the sequencing itself, but the approach to the project was quite different, okay? So this approach that you see here is the approach that was taken by the publicly funded project. So in that project, you've got 2 male genomes and 2 female genomes, okay? So 4 individuals. What happens is that you then start off, so I've already mentioned this, and you should very much remember that from the previous lecture as well. So you've got all this genomic DNA, you chop it up with restriction enzymes, and you put it into these back libraries. So basically you're cloning it into these bacterial plasmids. You then organize it, so according to your genetic markers. So remember, we've got a whole range of genetic markers, and we're aware of where these genetic markers are placed and what is the locus. So where is the location and position of these genetic markers? We already know that, okay? So we've got that information already. So on that basis, we can then start to organize and map these large clone contigs. As you can see here, so you can see we're now ordering them here, and you can see obviously some clones will have overlap, etc. So that is the contig of clones that you see here. And now before that is then sequences, you actually chop off, you use restriction enzymes again to actually restrict it or to chop off the DNA in these clones, and then you sequence it, as you can see down here. Once you have the sequences on all these bits of DNA that comes from that clone, you then assemble that, as you can see down here. Okay, so you can obviously see here that because of these overlaps here, so overlaps between clones and the contig, you get many overlapping reads of each segment of the original DNA. Okay, so you can see that here. So this bit of DNA here, this is an overlapping bit of DNA that would obviously be read in numerous times. So if you only sequence it one, you would obviously read this bit here twice, because it is found in both of these back clones. And that's really very much necessary, because that will overcome some of the difficulties and accuracy that you will need in terms of assembling the sequences. To assemble them, you need to know these overlapping sequences. And also, the Human Genome Project, most of the genome was not only sequenced one time, but actually there was a coverage of the genome about 9 times. So every single base in the human genome has been read or determined 9 times, and that's really called the coverage. Okay, so each base, the final sequence, has been present on average in 9 different reads. Okay, and this is just really showing you the map of the chromosomes. So we've got the genetic and the physical map. We've very much already spoke about that. So we've got all these genetic markers. Okay, we know the position of all of these genetic markers because remember, you make these genetic maps, remember they are made from families. Okay, so you look at these family trees to look at the heredity of these genetic markers. Again, thinking back to the previous lecture. Okay, so we've got the loci for all of these genetic markers, and based on all of these genetic markers, you then make this physical map, and you order all of your back clones into a contig, and that is really what you can see here on the right-hand side. So on the right-hand side here, we have got our contig. So it's really very much a question initially of getting all of these pages or to organize all of these clones according to their position really within the genome before you start the sequencing. Now this here really shows you the back clones which cover the cystic fibrosis gene, so cystic fibrosis transmembrane conductance regulator gene. I'm sure you have heard about cystic fibrosis. We're not going to talk about it in this lecture or the next lecture, but you will hear much more about cystic fibrosis next year, okay? So what this here just shows you all of the back clones which covers the cystic fibrosis transmembrane conductance regulator genes. So you can see there's a number of clones here. Now what you can see down here is really a set of numbers. So it really tells you where, in which library, on what plate, in what row, and in what column on this. So if you recall, looking back at the previous lectures that we did last week, that we had all these 96 will plates, and within these 96 will plates, that's where we store all of these different back clones in the freezer. And this here just is really an indication or a number for a particular clone. So you should somewhere be able to go in and read a number, which I really can't see, on these clones here. And if you wanted to look further at, let's say, this bit of the gene, then you, based on the number, so this clone here has got a number, and based on that number, somebody would be able to go and find that particular clone in the freezer and then post it out to you. Very well organized, you know, all of the clones are appropriate labeled and it can very quickly be found. So that really means you can look at particular sequences, you can look at all of these clones if you wish, etc., etc. Okay, guys, I'm just going to stop for 2 minutes because my son is trying to get me. So just give me 2 seconds. What's up, son? Okay, I'm sorry guys, my son was just heading off to school. He's doing history exam today, so he's just in the middle of all his higher exams, so he just couldn't find some things there. So please, I'm very sorry. But I'm back, okay, so I'm ready to go again. Okay, so as you will recall looking at the timeline, really many years were spent on this genetic and physical mapping and making these contigs before sequencing really started. And actually, before sequencing could start, so before you could start sequencing all of these contigs and all of these backbones, sequencing technology really had to be improved, okay? Because at that time, sequencing was a manual technique. We'll speak about that in a minute and look at what that actually means. But, you know, you very much had to stand at the bins and do all of these individual reactions and then run them out on a gel, etc., etc. So really, they needed a much more high-throughput sequencing technology before they could really start sequencing all of these back clones. And why they waited for that technology, you can see here, and we also saw that and spoke a bit about that on the timeline, other genomes were sequencing. So you can see here, influenza bacteria was sequenced. You can see the completion date, you can see the genome size, and you can see the estimated number of genes, okay? Now, larger genomes were also sequences. So the biggest one down here, as you can see, is the Drosophila fly. The Drosophila fly was completed in 2000, so really just before the completion of the human genome. And you can see estimated number of genes is about 13,000. Now, interestingly, you don't know that yet, but what we find in the human genome is that there's about between 20 to 25,000 genes. And you can see here that this plant, mustard plant, have also got an estimated number of genes of 25,000. Okay, so let's look a little bit at the sequencing technology, okay? So the most widely used sequencing methods for the last 40 years, really, is what we call the Sanger methods, okay? So that was a method which was developed by Frederick Sanger and his colleagues back in 1977. Now, what this technology is primarily based on is really the random insertion of a D-deoxynucleotide, or deoxyribonucleotide instead. So it's a D-deoxy instead of a deoxyribonucleotide, okay? And when that D-deoxy, so this is the DDNTP that you can see here, you can also see that depicted in the drawing up here. So when this one is incorporated, instead of the normal, so this is just one of the normal DNA bases, okay? So when this D-deoxy is incorporated instead of this normal DNA base, then the DNA polymerase does not work anymore and the reaction is therefore terminated, okay? So you obviously need DNA polymerase to keep building on these DNA bases along the sequence. Okay, so the bit of DNA that we are sequencing, you need DNA polymerase to put on these complementary DNA bases. But when it incorporates, this just happens randomly, when it incorporates one of these D-deoxynucleotides instead of the normal DNA base, then the reaction terminates. Okay, so it stops. Okay, so let's look a little bit closer at that here. Okay, so this is the manual Sanger sequences. Okay, so this is before we get on to high throughput sequencing, as we'll see in the next slide. So what happens here? So up here, we've got a bit of DNA that we want to sequence. So obviously, first of all, you denaturate that. So you get single-stranded DNA instead of double-stranded DNA. What you then have is one primer. So you can see that one primer. There we go. So you can see that one primer up here. And that one primer is labeled. Okay? It could be labeled, in this particular case here, it could be labeled with fluorescence, so different fluorescent probes, or it could be labeled with radioactivity. Okay? So different ways in which you can label it. Doesn't really matter here. But really, what is the point here is that it is the primer that is labeled. So you need 4 reactions tubes. Oh, I've lost my hat. There it is. So you've got the primer. The primer is labeled. And you have got all your DNA bases. Okay? So we've got the 4 DNA bases, as you can see here. You then split that into 4 tubes, as you can see down here. And then you can see that into each of these tubes, we add the D-deoxy ribonucleotides, okay? So the 4 different, okay? So obviously we've got the 4 DNA bases, and into each tube, we add one of the 4 D-deoxy ribonucleotides and DNA polymerase. So what happens now in the tube is obviously that the primer binds, okay, so the primer will bind to our DNA, and then the polymerase will obviously start to build on the DNA bases on the bit of single-stranded DNA that we are sequencing, as you can see up here. Now, by completely randomly, occasionally, instead of using the normal DNA bases, this D-deoxy base will be incorporated. And when that deoxy base is incorporated, the reaction will stop. So the result of that, as you can see here, is that you are going to end up with all these different lengths of DNA, and these are completely random, but as the reactions progresses, you should really end up with all combinations of lengths that you could find within the DNA, okay? So you can see that you should really then be able to order these. So on this particular strand that we're looking at here, you can see that we have got adenosine at 3 locations, okay? It gives us these 3 different pieces of DNA. You run that out on a gel, and obviously the smallest bit is going to be towards the top of the gel, and the longest bit and the smallest bit is going to travel further, okay? So the smallest bit is going to be down at the bottom here. So you run all of these reactions out for the different DNA bases, so for the different D-deoxy bases. And then based on that, you can look at the length of the different bands, and you can put together your sequence here, okay? So if we start 5 to 3 end, you can obviously see that the first base, so the first band is here, from this reaction here, and it's an adenosine. Okay, so that must be the first base. So you can then put together your sequence here, as you can see. So really the most important thing to remember here is that you have 4 reactions. Okay, it's not one reaction. It is actually 4 different reactions. And that these terminate at different points in the sequence. And on that basis, you can run it out on a gel, and you can then look at the length of the bands and put together your sequence. However, that would obviously take far too long, because imagine all of the many reactions that you would need to run to do the whole of the human genome. So instead, they nearly needed to develop a method which was much quicker as the one that we've just looked at. So that one is really shown here. So basically what you do here, same principle as before, but as you can see here, these D-deoxynucleotides, so these D-deoxy DNA bases, as you can see here, they're labeled with a different fluorochrome, okay? So we've got red, blue, green, and pink. So it's basically just a different fluorochrome. That means that you can run it all in one reaction. So you still start with a primer, but in this case, it is not your primer which is labeled. Okay, because that's why you need the 4 reactions, because if you put all of that in one tube, in the manual one, you obviously couldn't distinguish between the different bases. Okay, because what you actually detect is the primer. But in this particular case, it is actually the D-deoxynucleotide which you're detecting. So that is what is labeled here. So basically it works in exactly the same way. So this D-deoxy is obviously randomly terminating the reactions. So you have many reactions going on, and you will then end up with all these different length of change of DNA, as you can see here. And now once you run that reaction, you then run it on a capillary gel, as you can see here, and obviously with a laser, you activate the fluorochrome, and you can read you activate the fluorochrome, and on that basis, you can obviously determine what base or what D-deoxynucleotide has been incorporated in that particular position. Okay. You can then, so you can see all of this is really computerized, right? So you can see that that is then read by a computer here. And what you actually see, you get this chromatography down here. So this is really the laser detection of these different fluorochromes. And then the computer really put together the sequence for you. So you don't need to sit and read the sequence on a gel as you did in the previous example, right? The computer really generates this sequence for you. And here, so really, you got advances in the Sanger CNA sequencing technology, and that really significantly reduced the cost. Because you have to imagine that if you had to stand and do these manual sequencing reactions, imagine the time and the manpower that would require to sequence the full genome. So a reduction in cost very much with the development of this computerized technology. And here is actually each individual one of these boxes is a react. So in each one of these, you actually get sequencing occurring. So the sequencing will happen in each one of these boxes here. And then, as I already said, what then happens at the end very much is that the computer, based on the laser and the emission can measure what fluorochrome, and each deoxynucleotide obviously has the fluorochrome attached, and on that basis you can then read the sequence. And this is really what the reading, and the computer will then really very much put together that sequence for you. We've already really spoke about coverage, so that each base in the genome was read more than once. So each base, before the final sequence was actually published, was read on average 9 times. Also, putting together the sequences of all these contigs was done by a computer, so that was not done manually by an individual. The computer assembled all of these sequences into the contig. Okay, so this is really just a summary of the approach that was taken by the publicly funded Human Genome Project. So in this project, as we have spoken about, it was a clone-by-clone approach. So they did these contigs, they sequenced them, and then they put the contigs or the contigs together based on these sequencings, and particularly the overlapping sequences. Disadvantage is obviously this is a very slow process, and slow means also that it's an expensive process. So first of all, you could see in the timeline how long it actually took just to put these contigs together. So actually making genetic maps, then making physical maps, and then getting your contig. Very slow process, and obviously with slow typically means very expensive. Now, what is the advantage? The advantage very much is that this method is quite effective at getting over regions of highly repetitive DNA sequences. Also, another advantage is that you've obviously got this library in your freezer. So you're able to go back and retrieve any clone that you might want to revisit. You might want to look at something else with respect to that clone. Whatever anyone really wants to do, you can go back and retrieve that clone later from your library. Now, Celera used a quite different approach to the one which was used by the public, and the approach that Celera used is called shotgun sequencing. You already saw how in the public genome project they used 4 genomes, 2 male, 2 females. So basically 4 human genomes were sequenced. Now, in the Celera project, they only used 1 genome, and the rumor says, whether it's true or not, but the rumor actually says that this was the founder of the company, so Craig Venter, that it was actually his genome that was sequenced in this project. Now, the approach that they use is basically just to blast the genome into small fragments, and then they sequence all of these fragments. So they do not make back clones. They do not make genetic and physical maps and make these contigs. They basically just blast off or blast the genome into smaller bits. I sequence all of these bits, and then they use the power of computers to reassemble the sequence. Okay, so we can't put it back together on the basis of all these overlapping clones, etc. You can't go back and revisit. But when you take this approach that Celera took, they very much had to rely on the public databases of sequence and mapping information in order to assemble their sequence. Okay? So remember that the publicly funded project, they would publish within 24 hours, they published all of the sequence and mapping information that was available. So as the different mapping and sequencing data became available, it would be published, for anyone to see on the internet. And obviously Celera could utilize that because they could go in and find that bit of information. So if they had issues or problems with ordering some of their DNA sequences, they could really go in and get a lot of support from the public databases. So obviously there was a lot of competition, a lot of ugly competition between Craig Venter and Francis Collins, who led the project on the public side. And a lot of that competition, or ugly competition, was really because Craig Venter's philosophy obviously went completely against the philosophy of the National Institute of Health or the publicly funded Human Genome Project. So their philosophy was very much to share the data, should be available to everyone, while obviously Craig Venter's vendetta, or what he wanted to do, was very much to patent this information and basically to earn money on it. So the story goes, whether that's 100% true or not, I don't know, but the story goes that a mutual friend of Venter and Francis Collins invited them around for dinner, and they even said it was pizza. And over that dinner, they agreed to announce or publish the sequences simultaneously. So remember, there will be a sequence from the private, so Craig Venter's company, and there are sequences, so 4 genomes sequences from the nationally funded project. Now, at that particular time, Celera didn't really have enough sequence on their own, but remember, they've only barely been working for 9 months. So if you look at the different time scales of these projects, it is quite significant. So really, at the time, at this particular time, a readable genome was not really available from any of these 2 projects. However, what they did was that they agreed to put their separate feelings aside and their separate agendas aside, and then publish the sequence of the genome simultaneously, so at the same time. Now, this was really quite big in America, and this is just showing you that in June 2000, so all of this is in June 2000, so in June 2000, the White House announced, so you can see it's Bill Clinton here, announced that the majority of the human genome had now been sequenced. So there was a working draft, and at that time it was 80%. And you can see that on either side of Bill Clinton here, we have got Francis Collins and Craig Venter. And you can see that Bill Clinton here is saying, without a doubt, this is the most important, most wondrous map ever produced by humankind. So this working draft, 80%, was made available on the web July 2000. This was followed then by a publication of 90% of the sequence. And in July 2003, 99.99% of the sequencing was completed. This is just showing you in 2001. Okay, so this is where 90% of the sequence has been found. So here you can see the twin publications. So simultaneous publication coming out on the same day. One in Nature, so that is the public consortium, is publishing in Nature, and Celera is publishing their sequence here in Science. So back to back, simultaneously, these 2 publications appear in 2001 with 90% of the human genome sequenced. Okay, so the third phase, so the rest of it, I've only got very little left today, a couple of minutes. We're just going to start really the last phase. So once we've got the sequence, what does it actually all mean and what can we actually use it for? So that's the third phase. So a couple of minutes today, and then the last lecture really is very much about, you know, what is all of this? What does the human genome tell us? What have we used it for, etc. So the first phase, as I said, annotation of the genome sequence, and what does it actually all mean? And to some extent, we don't really know yet what it all means. We are making sense of it as we go along, and that is still ongoing. Okay, so we by no means made complete sense of the human genome yet. So what have we actually learned? So these are the 8 different points, or actually 7 points, that we are going to cover, mostly tomorrow. But also the last point down here, disease genes, we will very much cover next year, both third and fourth year actually, as you can see here. Okay, so we'll start off with, and we'll look at that today, what do we actually have in our genome? How do we compare to other species? What makes individual humans different from each other? I'm going to talk a bit about the fine structure of inheritance. We're going to talk about human history and look at the evidence for human evolution. And we're going to look at the way in which our gene controls our response to medication. So these are the ones that we'll be looking at today and tomorrow. So we're only going to do the first one today, really. Okay, so how many genes? Well, first of all, 20 to 25,000 genes, which was actually a lot fewer than they expected. So if you recall from the mustard plant, they found 25,000 genes. So it was quite perhaps a disappointment to many when they found that actually in the human genome, there wasn't any more than what was actually found in the mustard plant. So a lot fewer genes than what we expected. Genes actually only make up about 1.5 to 5% of the genome. It depends a little bit on what publications you read, etc. But between 1.5 to 5% of our genome is genes. The rest is what in the past was called junk DNA. And it was originally thought that this junk DNA really didn't have any functions. But today we very much know that that's not necessarily the case. So a lot of this junk DNA actually do have functions in humans. Okay, so this here shows you the anatomy of chromosome 16. And here you can see some of the major diseases which is associated with chromosome 16. So I'm not really going to go through them. You can look at them at your own leisure. Okay, but these are now the diseases that we know is associated to chromosome 16. So you could see here, so this is just an example of one of the chromosomes. So many of the chromosomes we now have the same kind of information available. So what do we know? Well, we obviously know how many base pair there is. So 90 million base pairs, just above 90 million base pairs. There is in chromosome 16. Single nucleotide polymorphisms. Okay, so we talked a little bit about single nucleotide polymorphisms. And this is something that you will revisit next year, because actually on the basis of the Human Genome Project, so on the basis of this sequencing of the human genome, so there's 5 genomes in total. Remember, 4 from the national project or international public funded project, and then the 1 genome from the private company. And really, when they looked at these, what became apparent was these single nucleotide polymorphisms. So these single basis which was different between people, okay? And on the basis of that discovery, a lot of larger projects was initiated which looked at these single nucleotide polymorphisms. But you can see here on chromosome 16, they have found nearly or just below 2 million single nucleotide polymorphism variants. Okay, chromosome 16, 805 confirmed protein coding genes found on this chromosome. 65 is putative proteins. 429 are pseudogenes. So what the pseudogenes means, these are basically gene copies that have lost the ability to code for a protein. So once upon a time, they did code for a protein, but they have lost that ability. And some of that might be due to these retrotranspositions. So we talked about this, these transposons in one of the previous lectures. I can't remember if it's the first or the second one. But, you know, this is basically these DNA sequences which can jump about in the genome, which they did a lot in the past. Less so now. Many of them are not mobile anymore, as we talked about. But you'll recall that we talked about vitamin C, so humans can't no longer make vitamin C, and that's basically because of the insertion of one of these retrotransplants into the enzyme, which is responsible for making vitamin C. A lot of RNA genes as well, as you can see here. So different types of RNA. So these are really very much involved in different functions. So ribosome function or splicing, etc., etc. So different RNA functions. But the point is that there is quite a few of these RNA genes, as you can see here. Now, what do our genes do? So that is what is shown here. So you can see that the majority, well, first of all, the majority is actually unclassified, okay? So these are genes which are unclassified. So we don't know yet what they actually do. So that might keep some of you in a job for many years to come to try and clarify these unclassified genes, or discover what these unclassified genes are actually doing and coding for. Now, you can see here that transcription factors, so a large proportion, biggest proportion, except for the unclassified genes, is transcription factors. So 12% of our genes code for transcription factors. So really the biggest group of genes code for genes which code for protein that are involved in regulating other genes. Okay? That is the biggest proportion of our genes are coding for transcription factors. Another big class of genes, as you can see here, 6.3% are coding for receptors. Nucleic acid binding, 8.5% down here. Transferases, 8.8%. Now you can have a look at the rest of these yourself. But it just shows you the different of what our genes are coding for. So what proteins are our genes coding for? Okay, so I'm going to stop there. And tomorrow we will start off with looking at comparative genomics and evolution. So I'm just going to stop sharing the screen. And then first of all, any questions before we finish? No question. And second thing, remember to do your workshop for tomorrow, okay? So you need to finish your workshop for tomorrow.
The other thing I quickly wanted to ask you about is your assessment. So I think your assessment is due on the 24th. Or somebody's joining us now. It's a little bit late. So your assessment is due on the 24th, okay? So obviously, only if there's anything you're in doubt about or you're not sure about, you can ask. But obviously, I can't really give you any answers to anything that's based on an assessment. But I'll be marking your assessments, okay? So I'll try and get those marked back to you pretty quickly after you